So for the project that we're talking about today, there were some challenges in getting started. One, like I mentioned, NRGene was a young company. Our breeding program was young, so we had heterozygous parent materials that we were working with. They were fixed for the autoflower trait, but they were heterozygous. And being that autoflower plants are generally small and have a short vegetative period, it was impossible to collect enough tissue from a single plant to get to be able to do the 30x sequencing. So we had to come up with a clever solution. So by putting together our heads with NRGene, we consulted with them, and we came up with this pedigree which could work around these challenges. So utilizing two of the parent lines that we had full sequence, fully phased genomes for, represented here by the two squares, the blue and the green, we were able to cross those to the same autoflower parent to generate the F1 progeny, which we would do the 30x sequencing on. And then we would select which of those F1s had the phenotypes that we wanted, cross those to generate 200 progeny that we would do 1x sequencing on. And the beauty of this design was that by using the same autoflower parent and having the full genomic information on the two parent lines that we used, we could then infer and decipher which haplotype blocks in the F1 and the progeny came from that autoflower parent. So this process took a bit of time. Typically from seed to seed, it's a 4-month period. So this first cross took about 4 months. In the second cycle, we had to first phenotype. So we had to grow out these photoperiod plants, select which ones had the contrasting phenotypes so that we made sure the progeny would segregate for the other phenotypes besides the autoflower trait that we were interested in, and then clone those so we could have enough tissue to collect for the sequencing, make the crosses, get the seed of the progeny. And then the phenotyping of the progeny had to be done in 2 steps as well. So we had to grow out the progeny under 18 hours of light initially so that we could identify the autoflower progeny. Once those were identified, we then cloned the photoperiod progeny, flowered out those clones, collected the phenotype data for other phenotypes that we were interested in, and overall this took about 16 months, and then we were able to take that data, hand it over to NRGene, and they did their magic